This lecture will cover the diagnostics for hematologic neoplasms. This will include chapters 28, 29, and 30. Now, flow cytometry was originally designed just to measure the physical properties of cells based on their ability to actually deflect light. With advancements in technologies, not only can we now measure those physical characteristics, we can also measure the chemical properties, and this allows us to be able to phenotype the various cells in which we're able to define the maturation stages of those cells. Flow cytometry actually uses light scatter, light excitation, and the emission of fluorochrome in order to give us various data from different cells and particles within the sample that we run through the analyzer. Now, all this data composed together allows us to measure the quantity of cells, the size of those cells, any sort of morphology, and any of the internal and external structures of the cells that we're questioning and trying to figure out exactly what they are. And what it does is it just speeds up the process. So where we used to have to look at cells and use special stains, now we can look at multiple cells simultaneously as it runs through the analyzer, looking at various perimeters for all of those cells at once. Advancements now allow us to use various fluorescent dyes, which when they're ran through the analyzer will actually emit various signals that when those particular dyes are bound to specific monoclonal antibodies, and then that allows us to be able to diagnose various hematological malignancies. So what we're going to do is just kind of talk about briefly some of the pre-analytical, analytical, and post-analytical phases of flow cytometry. However, we're not going to go into a lot of detail because you do touch on this in some of your other courses in more detail. Now, some of the samples that can be used for testing within flow cytometry include things like bone marrow, the bone marrow core, peripheral blood, spinal fluid, pleural fluid, fine needle aspirates, lymph nodes, solid tumors. And what we do with all of these is measure the various cellular characteristics to include things like the granular makeup of the cell, the surface area of the cell, and anything that's kind of internal to the cell. Your bone marrow and peripheral blood samples are typically going to be collected in some sort of tube with an anticoagulant, usually EDTA, and they're going to be processed within 24 hours to 48 hours, just depending on what type of anticoagulant you use, but of course the sooner the better. Now, your other fluids and samples, more specifically from your lymphatic type tissues, they're going to be placed in a sterile container for transport, and they're usually going to be processed pretty rapidly just to maintain that cellular structure and activity. Now, in addition to what it's collected in, the sample also needs to be transported properly. Anything that's not transported properly, it's not going to maintain that cellular viability to be able to show everything that we need for within our testing platforms. So for transport of your fluid type suspensions like your bone marrow and your peripheral blood, those again are going to be transported in some sort of tube that's going to contain an anticoagulant, heparin, EDTA, but most typically it's going to be your EDTA tubes, and of course they're transported at room temperature. And again, they're going to be evaluated pretty quickly, within 24 hours. Your solid specimens, these are going to include things like some of your bone marrow core biopsies, your tissue samples, things like that. And they're going to be submitted in some sort of culture media that's going to maintain that viability. It's going to be a sterile container, of course, and or it could be something that has a little bit of saline in it just to kind of maintain that moisture so those cells don't dry out. Now, as far as processing them, anything that's kind of in a fluid suspension, it's going to have very little processing required other than if you're making any sort of special stains or anything. But with flow cytometry, it's not going to really require a whole lot of processing. If you have solid specimens like your tissues and biopsies, usually you're going to use something to actually mince those cells up, some sort of grinder or scalpel or something like that, and then you'll do that step of processing in order to be able to allow it to go through the flow cytometry. But all specimens need to go through various quality checks and staining process to make sure that sample is adequate for what you're going to need for the particular testing that you're going to do. In order to understand how flow cytometry works, we first have to make sure that we understand what exactly we're looking for. So on the membrane of the cell, we have these various things called cell surface glycoproteins and glycolipid molecules. So this is what we know or term our surface markers or our antigens that we actually use to identify the cell. So each cell has various expressions of a very unique repertoire of surface antigens, and we see these at different stages of cell development, which allows us to kind of differentiate those different stages in the various different cells that actually contain those different surface markers. By the use of monoclonal antibodies, meaning that particular antibody is directed against one particular antigen that is actually on that cell. So by using those antibodies, it helps us to identify which antigens are actually present on those cells by using the flow cytometry methodology. As technology advances over the years, what has happened is there's actually been an international nomenclature that was developed to be able to help with the identification of these particular cells. And so what that's actually called is cluster of differentiation, and so this is known as the CD system. And this is where we get the actual CD marker term from, is that cluster of differentiation. And what the system does is it actually assigns a number to each identifiable blood cell antigen that's discovered along the way. So right now we have more than 400 CD antigens that have been identified for blood cells. So this system of nomenclature actually allows scientists and clinicians, laboratory practitioners, and your pathologists and everyone to kind of communicate using the same universal language for hematology, in which we can actually use these CD markers for diagnostic and therapeutic purposes. So flow cytometry uses hydrodynamic focusing, which actually allows the cell to enter into the center of this sheath flow, and then or either it's inside of a particular carrying fluid, and so once it enters into that sheath flow, the single cell itself will scatter light as it enters one or more of the various light path sources, and what's actually happened is there's an excitation that's going on. So fluorochromes are actually excited to a very high energy source, and so the optical detectors will actually convert that scattered or emitted light from those cells into various electronic impulses, and then that's sent to various detectors using optical filters, and then those pulses and signals are then processed within like a computer type system for various analysis for classification, sorting, separation of cells. And that's done just based off that detection system where it's looking for various forward scatter, side scatter, and then of course that fluorescence that it's looking for. So it kind of compels all this stuff together, looking for those specific CD markers, placing them into a specific category, and then from there you'll use those various CD markers to kind of narrow down what cell types you have, which aids in the actual diagnostics of trying to figure out what cells are present. In hematology, the cell populations that are identified by flow cytometry of interest to us are the hemopoietic cells. This is going to be including any of your early cells, your early stem cell lineages, your granulocytic lineage, which is going to include your myeloblast, promyelocyte, myelocyte, metamyelocyte, your bands, your monocytic lineage to include those blasts and permonocytic stages, and your monocytes, your erythroid lineage to include those early stages as well as mature. Same thing with your megakaryocytic lineage, all the way through your immature forms, all the way up through your mature platelet, and your lymphoid lineage, all the way through those early stem cells, up through those mature cells to include those B cells and T cells. And your textbook does include a couple of charts of interest for us that specify what those specific CD marker numbers are and their association with the various types of cell lineages. And so you'll get to become more familiar with these as we talk about some of the various mutations that happen along the way. This chart here is going to additionally kind of list those various CD markers and the function of them, as well as the cellular expression. So what flow cytometry is actually going to do as far as within the hematology department is it's going to help us analyze the various neoplasms that we're going to see. Now for the myeloid neoplasms, we're looking at some of these various neoplasms that we're going to discuss a little bit later, but we're looking at things like the acute myeloid leukemias with recurrent cytogenetic abnormalities, the acute myeloid leukemias that aren't otherwise specified, the myeloproliferative neoplasms, as well as the myelodysplastic syndromes. And then when we look at our lymphoid neoplasms, we're looking at things like our B lymphocytic leukemia and lymphomas, as well as your T lymphocytic lymphoma. So we're looking at those B cell and T cell lineages here. Now typically when you think about flow cytometry and using the various CD markers, we think about diagnostics for various neoplasms. However, flow cytometry has kind of advanced to the point that we're able to use it for other things than just diagnosing neoplasms. For example, it can be used to diagnose and monitor immunodeficiency states like leukocyte adhesion deficiencies, chronic granulomatous disease, and also just to kind of monitor the T cells within HIV patients to get that CD4 or CD8 ratio, which helps us with, or helps the clinician with being able to recognize the disease state and treatment, and as far as outcomes that could be possible for that particular patient. It can also be used to diagnose paroxysmal nocturnal hemoglobinuria. Some of the old methods actually use a red blood cell lysis methods to be able to do this test, but now with flow cytometry, we're able to look at some other things that kind of help diagnose that. And it's a little bit more efficient and sensitive to the actual disease process. You can enumerate stem cells, do tissue typing, cell cycle analysis. Flow cytometry is being used to detect fetal hemoglobin for those fetal bleeds, and monitor things like sepsis, doing molecular analysis and drug testing. So there's a lot of platforms where flow cytometry is being advanced upon. Molecular techniques actually enable a clinician to monitor disease progression during treatment, and this helps them to make the accurate prognosis and predict the various response to therapeutics. What molecular diagnostics is going to do is actually look at everything from a DNA, RNA type of standpoint, looking for those various abnormalities. So within hematology, there are 5 main areas that we'll look at and use the molecular testing. And so what we do is we can actually detect mutations, gene arrangements, rearrangements, chromosomal abnormalities, to use with that for diagnostic and prognosis purposes for various hematological malignancies. We can also detect and quantify the minimum residual disease to monitor various treatment options for that patient based off of the malignancy that they have. And we can detect mutations in inherited hematological diseases. And some of the newer techniques that are coming out is the pharmacogenetic testing, which is able to detect various genetic variations that affects how certain drug therapies respond and how your body responds to those particular therapies. And then, of course, identification of hematological diseases, various infectious diseases that can affect us hematologically. So actually looking at detecting those particular organisms that can cause those diseases, or microorganisms that can cause some of those various abnormalities to happen within those cells. Now we're not going to talk here about the molecular diagnostics and what all goes into that, because we will actually talk about that a little bit more in some of your other courses, like within immunology and microbiology. So human cytogenetics is a study of chromosomes, their structure, and their inheritance. So chromosomal disorders are classified as structural or numeric and involve the loss, gain, rearrangement of either a piece of the chromosome or the actual entire chromosome. So clinicians and physicians who are caring for patients of various ages can order various chromosome analysis or karyotyping for those patients that have various either intellectual disabilities, infertility, ambiguous genitalia, short stature, if there's been a fetal loss or miscarriage, or any sort of risk of any sort of genetic or chromosomal disease. And they also use it for looking at various cancers. So when we look at it from a cancer kind of standpoint or a hematological neoplasm, we're looking for specific structural rearrangements that are associated with the various distinct subtypes of leukemia that are, what we've typically gone by the morphological characteristics and clinical features. We can kind of add this extra component to it. So cytogenetic analysis of malignant cells can help us to determine the exact diagnosis, and it can often give us a little bit of the prognosis of that particular malignancy, and it allows the oncologist to actually determine what the appropriate therapy may be and treatment options, and they can kind of also monitor those treatment options and see how effective they are based off this. And so some of the applications that we've used that you may be familiar with, and we've already discussed in other courses or will continue to discuss, will be something like using the FISH methodology, which is a molecular cytogenetic technique that uses DNA or RNA probes and fluorescent microscopy to actually identify individual chromosomes and the targeted chromosomal loci. One of the other techniques you may be familiar with would be something like chromosomal microarray testing. So again, some of these things we're not going to go in depth with in this course, but just want you to kind of know how it is these techniques are applied when we're discussing some of the various testing, and once we get further into discussing some of these various disease states of these neoplasms.